In this module, we'll be learning about historical developments of microbiology as a source of knowledge. So in this, we'll be covering meaning of the term microbiology, different historical timelines and the contributions of several scientists and workers during this period, and finally the scope and future prospects. The word microbiology takes its origin from the term micro, which is of Greek language, that means very small in size. So without including any particular clade or any particular phyla, microbiology encompasses all those organisms which cannot be viewed with naked eyes. So it will include coccoid cells, rod-like cells, spiral cells, and as you can see in the last image, the bacteriophages which are lying over a bacterial cell. So it's all about size, and different categories of organisms fall into different size lines. For example, the nanobacteria, which is most recently been discovered, falls in the range of 50 to 500 nanometer, while the smallest accepted bacteria, that is Mycoplasma genitalium, is around 300 nm. The largest virus that we can talk of, virus, which is a causing agent of several deformities in amoeba, that is around 400 nm. And a marine bacteria, Pelagibacter, is around 500 nm. The most prevalently and frequently talked about our own Escherichia coli, that size is around 2000 and 6000 nm. And for viewing all this, we need a microscope or any aid which can help us do that. So the major groups those are included in microbiology are bacteria, which are prokaryotic, meaning they do not have a outlined nucleus, so their genetic material lies freely in the cytoplasm, and their cell walls are made up of peptidoglycan. Very close in resemblance to bacteria is the group called archaea, which is prevalently found in extreme environmental conditions, and their cell walls lack peptidoglycan. Now this is what differentiates archaea from bacteria. Next group similar is protozoa, which is unicellular but marks its difference by being the aerobic eukaryotes. So they have complex organelles, and from here begins the division of labor at cellular level. So there are different organelles which are dedicated to a particular task. As a common feature, they all obtain their nutrition by absorption or ingestion. Next group is algae, which marks several organisms, all eukaryotic, ranging from unicellular to multicellular. They possess distinct cell walls, which is made up of cellulose, and like protozoa, they are also polyphyletic in origin. That means they do not have a common ancestor. Different organisms lying within this group, they fall or trace back their origin to different ancestors. These can be photosynthetic, as we know about green algae, cyanobacteria, or they can also obtain their energy by utilizing organic molecules prepared by other organisms. We also include fungi in this, which are again eukaryotic and have multicellular cells, but their cell walls are distinct from all other groups in the presence of chitin. These are considered the chief environmental decomposers because they can give out enzymes and dissolve things. That's a form of nutrition most of the fungi have, or they can absorb nourishment. So they have a very important and key role in the nutrition cycles. Another interesting group is that of helminths, which is again a wide group that includes all flatworms and roundworms that are pathogenic. So these are also not any one particular class. In addition, they are also large enough for viewing, but the purpose we include them over studies here is that a part of their life cycle, mostly the sexual spores that they produce for being transferred from one host to other, those are microscopic in form. And the last group that we include here are viruses. These are acellular and they have a nucleic acid core, which may be a DNA or a RNA, that is their genetic material, and it is surrounded in a protein coat. We call them acellular because outside, lying in the environment where there is no host, they do not show any biological activity. But as soon as they can find a host and enter inside it, their biological properties start. They start reproducing, they start using the host machinery in their favor, and start making their own genetic material and proteins. Studying microbiology is very important to understand the crux of evolutionary existence, because the evolution began with the evolution of microbes. And to understand the physiological and biochemical processes that lead to evolution, we also need to understand the working and the functioning of these microbes and their interactions with the environment. Second important aspect of the need to study is harnessing their enzymatic properties. These microorganisms have a plethora of enzymes, and if we can utilize those enzymes for our purposes, it can be revolutionary, and it has proven to be revolutionary in the fields of food production, agriculture, bioremediation, and several other ways. Although very few microbes are pathogenic, but the havoc they create is enormous. And to control those diseases, we need a very good understanding of their biological properties, their life cycles, their pathogenic cycle, so that we can manage them. And again, the management of these diseases comes through applications of antibiotics and vaccines, which again depend on our knowledge of microbial world. In the last, we also move towards the genetically engineered organisms, where the genes are mostly taken from microbial world. Then we use them as a vector for transferring. Even if we are genetically engineering some plants or animals, the vectors are mostly prokaryotic. And then even for the expression sake, for checking and all, we again need microbes. So it is very crucial that we understand their functioning so that we can utilize them for our benefits. The entire evolution of microbiology as a subject can be divided into four different time scales: the discovery era, the transition era, the golden era, and the modern era. So it all began with the prevailing thought of Greek philosophers, Aristotle being the most important advocate of those times, who said and believed that any non-living material can give rise to living organisms. So that was the Aristotelian thought, and he had great influence on the society. So he had huge followers, and this particular sect is termed believing into the theory of spontaneous generation. Then, for the first time, around 466 years before, that is 1546, a thought came, a discussion began on contagious infections. So, that was the first time that some of the properties of microbes were taken into consideration. Then, in 1676, a curious person called Antonio von Leeuwenhoek, he invented microscope, and he wanted to view what all was around, and that began an era of microbial studies. So in the discovery era, we had a group of scientists who had a curiosity to see what were the several organisms or several non-living things made up of. The first among those lines was Robert Hooke, who in 1665 took a piece of cork and viewed it in a microscope that he had made. He could see hollow cells over there. So that was not exactly a microbe, but yes, it led to formation of a microscope and an integrity of seeing what was not visible to the eyes. With that, he started viewing other objects also, and then he moved to the biological world, where he illustrated fruiting structures of molds. So he formed a book, he formed all his observations into a book called Micrographia, which gave light to several other workers working after him. In 1858, Rudolf Virchow had a thought that all the cells could come from pre-existing cells only. So together these two concepts, they formed the cell theory that said that all living components are made up of cells. So the basic unit is cell, and all these cells can come only from pre-existing cells. So this was in confliction with the theory of spontaneous generation. In due course of time, Leeuwenhoek, a normal grinder, got extremely influenced by Robert Hooke's micrographia and developed his own microscope to observe several living, non-living components around him. He is also therefore considered father of bacteriology and protozoology because of several observational descriptions that he gave in the years to come. He made a very simple microscope, a crude kind of instrument, but he was so much into viewing what all was around that he used his own teeth scrapings of other persons, rainwater, even pus cells of infected ladies, so that he could see what was lying inside that. And he saw several moving structures. So he called all these animalcules. And he was very scientific in his approach that he drew exactly what he saw. So all these drawings, the bacterial drawings, those were communicated to Royal Society of London and almost created a revolution in the microbiological studies. Transition era was the time when the debate between the spontaneous generation and biogenesis got heated up. Spontaneous generation that was advocated by Aristotle and his followers, it had several hypotheses. All were based clearly on observations, as they saw that wheat lying in a storeroom that had rats after some days, a shirt full of sweat that attracted flies. So all these observations gave them a cue, and they started believing it, that these non-living organisms such as wheat and the shirt, they could give rise to living organisms. So they believed that the essence of life originated from another essence present in the non-living materials, which they called vital heat or pneuma. Contrary to this, a sect started being built up that believed that living organisms can arise only from pre-existing life and there is nothing called pneuma or vital heat. Main participants of this debate included Francesco Redi, who against spontaneous generation showed that maggots would not arise from any decaying material lying around. Then John Needham, he opposed Francesco Redi and came in support of the spontaneous generation theory. Finally, in these series of events, Spallanzani showed with a series of experiments that if sterilization is done properly, then any non-living broth or nutrient material that cannot be clouded with microbial growth. Now, beginning with the contributions of Francesco Redi, he was a keen scientist and performed several sets of experiments. His interests led him to conduct several studies on microscopic organisms, and he also worked with lots of insects. His studies got published in the form of a book called Experiments on the Generation of Insects, and he also very precisely described several parasites. Now, those parasites, all which were pathogenic on humans and included Fasciola and Ascaris, and because of all these contributions, he was considered father of experimental biology and father of modern parasitology. Regarding this debate, he had a curiosity of understanding, can meat give rise to maggots? So if there are flies and if there is meat lying around, we see another set of maggots coming up. So what if there is no meat or there is no flies around the meat? Even then, can there be maggots? So he formulated a hypothesis of his own that if meat creates maggots, then wherever meat is, there can be maggots. To verify his hypothesis, he set up an experiment. And this was for the very first time that the concept of control was included. So he first took a flask which was not covered by anything. It had meat inside it. So he treated this as control because this was what was happening around. And then he made his experimental setups where he kept a meat in a jar which was covered tightly with a cork. He had another jar which had meat inside and it was covered with a gauze. After some time, after few days, He kept on observing his experimental setups, and he found that, as expected, the control that he took, it had maggots into it. Second, where the meat was precisely and controllably sealed with the cork, there were no maggots around, because even the smell or anything of the meat could not move out. And in the third experimental setups, where there was a gauze, meat was very clear. It did not have any maggots around, but small flies, they kept coming and assembling on the gauze. So, in his very scientific approach, as we can see, he first had a question that he needed to answer, he formulated a hypothesis, and then he conducted experiments and observed the outcomes very precisely. Now, in the last part, he had to gather his results and put them to analysis. So his results were like, he said, open jars, there were maggots, and the analytical question that came was that from where did these maggots come? Second set of observations led to results, jars covered with fine nets, there were no maggots, and the analytical question was, what was the purpose of the sealed jars? So the wider question, spontaneous generation or biogenesis. So with these analysis, he was very clear that meat was not giving rise to maggots, because if it were so, then maggots should have been present everywhere. And second, maggots can come only from maggots. So his precise answer to the question was that life can arise only from pre-existing life. So, apart from his contributions to raising this debate against spontaneous generation, his very important contribution is development of a scientific aptitude. For the first time in the history of knowledge and understanding, experiments were being formulated, experiments were being conducted, and then observations were being taken and analyzed. And this is why he is considered as the father of experimental biology. As the thought of a spontaneous generation prevailed for many centuries, it was very deep rooted. It was very hard for people and the followers to accept that it was wrong. So came up a person called Needham, who was a Irish priest, and he challenged Redi's observations and analysis. For this, he put up an experiment where he boiled a microbial broth to sterilize it. Then, after some time, he covered it, and he still observed microbes growing in that broth. So, his primary question was, from where did the microbes come? If there was no life without life, after sterilization, every form of life would have been disappeared. There would have been no other source of cells. So, there should not have been any microbes. Now, if microbes are there, this means that the broth had some pneuma or the vital heat which could give rise to microbes. So, this was his justifications towards the theory of spontaneous generation. After Lazzaro Spallanzani was another contemporary Irish scientist with very keen observation and a very defined set of analytical skills. His contributions to biology on the whole are very immense. So he not just worked on microbiology, he gave lots of contributions in the fields of physiology, reproductive biology, developmental biology. So for the first time, he was the one to show that the mammalian reproduction, it requires semen and ovum. And he didn't stop there. He went on to perform in vitro fertilization. He also performed artificial insemination, and he also gave the concept which was there about regeneration, because he said that there are several animals, newts were the ones he observed very closely, which when inflicted with some damage, they can regenerate some parts of their body, limbs and tails, etc. But one of his major contributions to physiology is his understanding of digestion. Till then, it was believed that digestion is a mechanical process. So he came up with the idea and established it that it's not just mechanical grinding that leads to digestion of food. There are several digestive enzymes, there are body juices which work at particular conditions within our body. And so, digestion is more of a chemical process than the initial mechanical one. So, apart from these, his contributions to the debate of spontaneous versus biogenesis were that he reperformed and refined Needham's experiment to validate his results. So he took four setups. In the first one, he had nutrient broth, a culture media in the flask that was open, and he observed that after some time there was microbial growth into it. So he had the thought, the question was whether it is a spontaneous generation or biogenesis. In the second setup, he corked the flask carrying the nutrient broth, and then he saw after some time that there was microbial growth into it. So again the question was that from where did these germs come? In his third setup, he had this broth in the flask covered with the cork, and he heated it. After heating, he opened up, and then after some time he saw that there was microbial growth into it. So it clearly stated that it was air that carried the germs to the culture medium. Otherwise, after sterilization, there was nothing else which could have got into it. In his fourth setup, he boiled the culture media, he corked it during the process, and he did not release the cork. So the medium did not come in contact with outside air. And finally, he saw that there was no microbial growth into it. So he concluded that heating can sterilize the air that carries germs. This was a major blow to Needham's experiments and his support to spontaneous generation, and therefore he was the first scientist to have clearly stated that life can only come from pre-existing life, so biogenesis needed to be more established. But to this thought, there were again opponents who claimed that the heating process was so severe and so long that the vital life force that the spontaneous generation advocates called pneuma, it got removed, it got killed, so that new life could not arise from it. So then followed a series of workers who came up in support, who came up in opposition, but at least the scientific understanding and the scientific approach became prevalent after these sets of experiments. So we had Schultz and Schwann, who were German scholars, and they were also in support of Spallanzani. So they took this view seriously, that it is the air that is the source of microbial growth, and so they passed the air coming to the broth through some strong chemicals, acids, or heat, and then they observed that their broth remained free of the microbes, and proper sterilization could actually help control microbial growth. Similarly, Schroeder and Von Dusch, they were the first to introduce the idea of cotton plugs. So that was the main thing that could hide the outside air in coming in contact with the broth. And then Darwin came up with his book, with his thoughts, that had this microbiological concept that diseases may be occurring because of some biological phenomena. So that was the basic trigger that included the idea that it is not some magic or anything or coming out of non-living things. It is basically a biological phenomena, and with this idea people started viewing microbiology as a source of understanding that could create an insight into the disease management.